Hey everyone, and thanks for joining me today for episode 842 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and I'm looking forward to speaking with our guest, Dr. Trudy Oliver, today. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom purchase orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And Trudy has some fantastic stories to share with all of us about herself and her lab. So get ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Trudy Oliver. So, Trudy, welcome to the show today. How are you?
I'm wonderful. Thanks for having me.
Well, thank you so much for joining us. And before we dive into the details of our interview, I'd like to take a moment to give our listeners a little bit of insight into your background and how you got to where you are today. So listeners, Trudy is a professor in the Department of Pharmacology and Cancer Biology, as well as a Duke Science and Technology Scholar at Duke University. She received her bachelor's degree in chemistry from Oklahoma Baptist University and her PhD in cancer biology from Duke University. Afterwards, she conducted postdoctoral research at the University of North Carolina, Chapel Hill, and subsequently at MIT. Trudy served on the faculty at the University of Utah for more than a decade before joining the faculty there at Duke, where she is today. And Trudy has received numerous awards and honors for her work, including the Heine Hansen Lectureship Award for small cell lung cancer from the IASLC World Lung Conference, the William C. Rippe Award for Distinguished Research in Lung Cancer from the Lung Cancer Research Foundation, the Lung Cancer Discovery Award from the American Lung Association, the American Cancer Society Research Scholar Award, the Damon Runyon-Rachleff Innovation Award, and the Jimmy V. Scholar Award from the V Foundation for Cancer Research. And Trudy, today we're excited to get to know you as a scientist, of course, but also get to know more about your life outside of the lab. So can you tell us, what do you like to do when you're not doing science?
Yeah, well, when I'm not in the lab, I like to be outside going for walks or jogs, and that gives me some time to think. I'll usually put on music or a podcast. I happen to like crime podcasts. And then I have two orange cats, Basal and Tuft. I'm sure you'll figure out why they have those names as we continue. And just, spending time with friends and family.
I love it. Well, it's great to hear a little bit more about how you spend your time. And you mentioned you've got these two cats. Is there a story behind them?
Yeah, actually, I had two other cats who I lost this past year, and friends talked me into adopting two baby kittens, and they're totally just a joy. I've never had kittens before. There's so much energy and just they love to play. And I named them after my two favorite cell types in the lung, and that's the basal cell and the tuft cell. So now it happens when I'm at work and I'm actually talking about basal cells and tuft cells, and other people are using those words. Every once in a while, I'll have a moment where I think they're talking about the cats.
I love that. When you first said basil, I was thinking the herb, but yes, basal cell makes more sense.
Yeah, exactly.
Well, Trudy, it's wonderful to hear more about your cats, your life outside of science, but I'd love to talk about your work as well. We sort of hinted at it with the naming of your pets, but how do you describe what you do to someone who is outside of your field or outside of science?
Yeah, I normally tell people, that I work on lung cancer. And when we get a little bit deeper, I explain that we're sort of obsessed with the fact that cancer can evolve or shapeshift. We study this type of lung cancer called small cell lung cancer, which is really the poster child for the ability of cells to shape shift or undergo what we call plasticity, and they have the ability to change from one type to another. And they have very different therapeutic vulnerabilities when they change. And so this is part of why it makes cancers so difficult to treat, why one therapy is not a one size fits all. So yeah, that's what we're fascinated with.
Wonderful. Well, I'm excited to get into some of the details of your projects as we go through our conversation today. But I think it's important to talk a little bit about motivation. I think life as a scientist is not always easy. So it's helpful to have little quotes or sayings or just mantras that keep you motivated. So do you have a particular quote or a saying or something that really inspires you?
Maybe a couple. I have this sign in my office right now that basically says, "We can sleep when we're dead. Drink more coffee." And that keeps me motivated to keep going. Also, I like to tell my lab it's just as hard and expensive and difficult to answer an unimportant question as it is to answer an important question. So we often use that to try to keep our focus on, well, if we're going to give our lives and time and effort and money to a specific problem, let's make sure it's the most important problem we can think of.
Well, Trudy, I love that mentality. I think it's such a good point for science because I think it's easy to sort of fall into this trap of the next incremental step in research as opposed to focusing on these big, very difficult problems.
Yeah, exactly.
Well, wonderful to hear about these motivational quotes, but I think oftentimes people you encounter can be quite motivational as well. And as a scientist, there's no shortage of fantastic collaborators and mentors and role models. So when you look at your own career path, are there particular individuals who you think really helped you get to where you are today or who you've looked up to over time?
For sure. I think there have been a lot of them, actually. I was thinking back when I was in college, my mom and dad didn't really go to a 4-year college, so they couldn't really advise me on certain types of things. But my mom did encourage me to go to college. And when I was in college, an advisor encouraged me to go look for these sort of summer research programs where I could get more in-depth research because I was at a very small liberal arts type of college that didn't have hardcore scientific research. So Dr. Mark Hemmerich encouraged me to find these summer programs, and that led me to the University of Colorado, to Paul Bunn's lab, where Barb Helfrich kind of became a, I would say, lifelong friend and mentor. She was really the one that encouraged me to go to graduate school and find PhD programs. And that's really what led me to find Duke, because I was very interested in cancer biology. And at the time, there were very few PhD programs in cancer biology, but Duke had one of them. So yeah, then I came to Duke, and my PI mentor, who was Dr. Rob Lexorea, was really fantastic and energetic. And then my postdoc mentor is Dr. Tyler Jacks at MIT, and he still stands out today as probably one of my greatest role models, just a phenomenal scientist and human being. And he really created this inspiring environment at MIT that I try to recapitulate as best I can in my own lab now.
Well, it sounds like you've had a wonderful constellation of mentors over the course of your career, and I absolutely love that. I'm curious, though, where your interest in science began. You mentioned that your parents didn't necessarily have this scientific background. How did you first start getting interested in science and start maybe thinking about this is something that you want to do for a career?
I mean, I found some notes a while back that even when I was a tiny kid learning how to write, I was saying things like, I wanted to be a veterinarian or a heart doctor. I also put that I wanted to be a clown.
Aww.
Slightly different, but I think I said I wanted to be in a circus. But then I realized that childhood dream because we had some kind of school circus and I got to ride a unicycle. So I feel like I checked that off the list early. And then that left all these other scientific ideas that I had. Mm-hmm. But really, I would say when I was in college, I had 2 family members, one on each side of the family, develop cancer. One was an uncle who had a brain tumor and he died very quickly. It was about a month, I think, from Thanksgiving to Christmas. And then I had a great grandmother on the other side of my family with a type of lymphoma, and she got chemotherapy. And I went with her to one of her chemotherapy sessions where there were all these people sitting around the room with these sort of bags hanging of chemotherapy sort of dripping into their IV. And it really just struck me. They were people of all ages, all different types. It was very obvious that like cancer doesn't discriminate. And that had a really profound effect on me. And it was sort of the beginning of thinking like, I really want to be an expert in this. And what does it take to become an expert in cancer? Like, I want to understand this disease.
Absolutely. And I think with a lot of these disease researchers, there is this sort of sad personal story or connection that goes along with it. But I think that can be tremendously motivating. So did you have a sense of how to take it from this idea of, okay, this is something I want to do, to like what steps you needed to take to get there?
Not at all. And that's why those mentors were really critical in my life, because I knew that I wanted to understand it, but I definitely didn't know what to do next. So that was when my college mentor, Dr. Hemricks, told me about summer programs. And that's how I found Barb Helfrich. And she was the one saying, you should get a PhD. Because to be honest, at the time, I didn't know the difference between a PhD and an MD. I had no idea what this would entail. And even when I joined Duke for the PhD program, I didn't really know what I was getting into. I remember distinctly getting excited, when do we get our textbooks? Because I thought I was just going to come and learn everything from the textbook. And I was really shocked to find out, oh, when you're in graduate school, you don't learn from textbooks. You learn from primary research papers. And you're going to actually do research in the lab to create primary research papers. And that's how textbooks are ultimately written. So I really feel like every step of the way, I was figuring things out as they came.
Oh, absolutely. And it sounds like that summer research experience in Colorado was a pivotal moment for you. What was it like for you the first time you were able to get into the lab and actually do research?
Yeah, I mean, it was very exciting. I think if you asked Barb, she would probably tell stories of me like breaking the pipette and all the things that I did wrong and had to learn the hard way. But I had no shortage of passion and motivation. I think back at that time, and we didn't have the TV shows where you can see the equivalent of CSI or whatever. We didn't have that when I was that age. So it was really something I had never seen before, and sort of blew my mind that you could do these experiments and have all the controls and get answers that were reliable and reproducible. And so I was hooked.
Oh, absolutely. And I think going from this point where you've sort of committed to the career to actually getting to the point where you are today is a long and often difficult road. Can you share, Trudy, what were some of these key moments along your journey that really helped you get to this point where you are a successful professor at Duke?
Being in science is a hard road. There are a lot of challenges, I guess I would say. You got to get used to different forms of, I would say, rejection and being resilient and to continue trying. So, for example, we submit papers and grants all the time, and there's lots of rejection and lots of critical feedback. But I think you sort of learn in that process to have a thick skin, don't take things personally, and realize that feedback is really a gift. Whatever someone sees, it isn't exactly what you wanted them to see, is an opportunity for you to keep refining your ideas until you can convince people to see things the way you see them. So I just think back when I was a grad student, sitting in seminars and things where I had no idea what they were talking about. Everything sounded like a different language to me. But I just tried to keep showing up and keep trying and keep thinking. And over time, step by step, I made improvements. I caught up with my peers. And I think it's just persistence, grit, resilience is kind of what gets you there over time.
Absolutely. And as you were ready to finish your PhD, there's often a big transition in terms of figuring out what you want to do next, where you want to go for a postdoc, or whether you want to do something else entirely. So for you, how did you make that decision?
I remember each of those milestone moments being not totally sure what was next. I think you meet some people who are just totally convinced of what the next step is. But for me, I always heavily debated, what do I do now? I remember moments at the end of my PhD where I thought, maybe I want to be an FBI agent. Maybe I'm just going to totally change my mind here.
So it's crime podcasts, right?
Yeah. And then I saw the fitness requirements and I was like, maybe not. Too many push-ups. But I remember at the end of grad school wanting to do a postdoc with Tyler Jacks, who I had met at a scientific meeting. one of these conferences. He was a top choice of labs that I applied to. And when I was admitted into his lab, I was very excited to go to Boston and experience that there. But even in the beginning of joining his lab, I totally thought he made the first and only mistake of his life in letting me join the lab. I didn't think that I was ready, and it was very hard and very challenging. But by the time my postdoc was over, I feel like I had demonstrated that I did belong there. But then again, at the end of the postdoc, I had another one of those moments of self-doubt. I don't know if I'm going to be good enough to go on the job market and get a real faculty position. But I had a friend say, if you don't try, you're definitely going to fail. And if you do try and it doesn't work out, is that really the end of the world? You'll just adapt and do something different. And so he totally took the fear out of it for me and gave me the courage to just go for it, go for a faculty position. And that's when I actually had like 6 different job offers and ultimately chose to go to Utah, which was a very beautiful state, amazing people. Was so glad that I took the chance.
Very cool. So then what ultimately brought you back to Duke?
Well, after being at Utah for about 11 years, I kind of was just needing a change. If you think about it in science, a lot of our careers are these little 4 to 5 year periods. You've got your undergrad for 4 years, PhD for 4 or 5 years, postdoc for 4 or 5 years. So I think there was almost like a program in my body of nothing's changed in a while. And then COVID happened and everybody was stuck in their homes thinking about things. And I think I just had this big itch and need to change things up. And I had had such a good experience at Duke as a PhD student that I decided I wanted to investigate what it would be to come back here to this part of the country. And everything just kind of worked out.
Well, I love to hear that the story had a happy ending. And I know you've had some remarkable research projects going on in your lab over the years. So I'd love to talk about your work more. Is there a particular project, Trudy, that you are working on right now that you are just the most excited about?
When I mentioned earlier that we are really into small cell lung cancer, and it's this type of cancer that can evolve or shapeshift. There's about 4 different types of small cell, and our lab has been sort of a pioneer in showing that these different types can change from one to another. And one of the least understood but most aggressive types of small cell is called tuft-like small cell. Not named after my cat, but the other way around. And it's called tuft-like because we have these cells in our body called tuft cells that have these little appendages called microvilli on the top of their surface. So if you think about it, it's like a little tuft of hair on the top of their heads. And those little microvilli are there to sense pathogens. Normal tuft cells will sense and respond to things like parasites. But the cancer can actually adopt a tuft-like state that does many of the things that normal tuft cells do. And as I mentioned, it's the tuft-like state of small cells particularly aggressive, so it's associated with some of the worst outcomes in response to chemotherapy and chemoimmunotherapy. But we don't really currently have any therapies that are specific for this state. And our lab recently found in unpublished work a therapeutic vulnerability for this state that looks really profound in a lot of model systems. So using human cell lines and mouse models, we see that the tuft-like state is really vulnerable to this particular target. So we are really excited to keep pushing this research and hopefully get it submitted in the next couple months and get this out there because we think this could be really beneficial for patients with this type of cancer. And it's not just limited to small cell of the lung because other neuroendocrine types of tumors found throughout our body, including in the nose, prostate, bladder, gastrointestinal tract, etc., they can all have these tuft-like states. So we think this might benefit a lot of people.
Well, Trudy, I think that is remarkable, and it sounds like a very exciting time in your lab right now.
Definitely.
And I know that in order to make some of these exciting discoveries in science, you have to have the right tools and supplies in the lab. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of ELISA kits, which offer high sensitivity, specificity, and can be used for a wide range of applications. Innovative Research ELISA kits are manufactured in their lab in Novi, Michigan, and are shipped directly to your location. They are a valuable tool for researchers and clinicians who need to detect and quantify specific biomolecules. Visit www.innov-research.com. That's www.innov-research.com to view their entire line of ELISA kits. And returning to our conversation with Trudy, I know you got to share some of the wonderful work that your lab has been doing recently, but we also hinted at some of the struggles that you've had over the years, whether it was these rejections that you mentioned earlier, the critical feedback that you just sort of have to learn to take as a scientist. I think not everything is these wonderful, exciting projects that always work out. So we love talking about the challenges, the struggles, the failures in science, because oftentimes these stories just never see the light of day. So Trudy, do you have an example you could share with us from your own career, something you really struggled with or a major failure that you experienced? And if so, could you walk us through how you got through that difficult time?
Well, I kind of think like I'm a scientist, I'm always struggling.
Mm-hmm.
I'm definitely struggling right now with, we're living in this, I would call it a sort of manufactured funding crisis. And so getting funding right now is very challenging. We had a grant that was a top 8% score that in normal times would have been funded. And then this year, as they changed the rules mid-game, it's looking like this grant's not going to be funded.
Oh, no.
So that's been very disappointing and caused us to try to scurry and scramble and look for new ways to get funding. So we're kind of in the midst of that right now. And yeah, it's challenging because I think that the job was already hard before these new conditions, and now it's even harder.
Absolutely. It sounds like a roller coaster of emotions where you get the review back and you see, oh gosh, 8%, that's looking promising. And then it's sort of, like I said, a roller coaster. How do you deal with that situation, or what really helped you not give up?
One is like, we have a pretty good sized lab, but last I counted recently, we have almost 20 different people working in the lab, and these people depend on me for their livelihoods. So there's definitely just this sense of responsibility of like, we can't give up. We have to keep working because we've got all these people depending on us. And the science is really exciting right now. I think this has sort of been the peak of my career so far. So I want to see this work go forward. And so we're just going to keep pushing. And hope is not really a plan, but we have a little bit of hope that maybe if the public learns more about the struggles that scientists are facing right now, that maybe Congress can come to a bipartisan agreement to make things better and allow us to keep doing the work.
Absolutely. Well, I appreciate you walking us through some of the difficult times that your lab is going through now. And I imagine you are not alone in this. So I think many of our listeners can commiserate and get inspiration in your motivation to continue. But we don't just want to talk about the difficult times. I think your lab has had so many successes over the years that we would love to celebrate as well. So Trudy, do you have a favorite success story that you would like to share, whether it was a big win or even just a small but particularly meaningful one?
Yeah, I think our most recent victory is a really big one for us. We published a story in Nature. It was the first time our lab has ever published anything in Nature, but really the science behind that story that just came out in September is really exciting. So I talked a little bit about small cell earlier. This is a tumor that historically the world thought arose in a neuroendocrine cell because it is a neuroendocrine type of cancer. And some model systems suggested that this tumor normally arises in neuroendocrine cells. But we were sort of bugged by the fact that when we introduce human relevant mutations into neuroendocrine cells in animal models, the tumors would never quite faithfully recapitulate what we see in the human disease. And that bugged us. We were trying to, one, create the Tuft-like state of small cell lung cancer, and we never could get it when we were using neuroendocrine cells. So long story short, we started investigating this basal cell, which is a stem cell in the lung that is known to normally give rise to both neuroendocrine and Tuft-like states. And when we changed our model system and started using the basal cell as an origin, suddenly we were getting tumors that looked and behaved much more like the human disease. And when we would put mutations relevant to the human disease into the basal cell, it would give us what we predicted. And the neuroendocrine cell didn't do that. So to kind of summarize, it really seems like the basal cell is the more likely cell of origin for small cell lung cancer. And that has really changed how we think about the disease, certainly how we model it. And I think ultimately is pointing us in directions of how can we perhaps prevent cancer earlier when it's initiating in these basal cells. And some other recent studies, completely independent from our group, people that took totally different approaches to this problem, using machine learning types of approaches, analyzing human tumors, they actually arrived at the same conclusion that we did. So that was a surprise for them that their data was implicating the basal cell and not the neuroendocrine cell, but it just sort of perfectly coincided in time and conclusion with our work. So that was very satisfying for us.
That is so exciting. And I think, like you said, lends credibility to this idea that your lab is putting out there. And this was not a small endeavor. Can you talk about just the scale of this? This was a lot of samples that you looked at.
We really took a plethora of different types of approaches, one using a lot of different genetically engineered mouse models and organoid-based models to arrive at these conclusions. But then we also partnered with a company who happened to have the largest data set we could find of human small cell lung cancer samples. So we were able to validate our findings and predictions using this very large human data set that really bolstered the findings.
Very cool. So how did your lab celebrate when the wonderful news came out?
We've not celebrated enough. Probably if my lab hears that, they'll probably be like, yeah, Trudy, when are we celebrating? We've had some small moments, but we actually haven't yet had a party as a group. So we need to do that.
Yeah, block that off on the calendar. I think the first Nature paper deserves a party.
I know, right? This is how it is. There's always just some fire to put out. So it's coming. It's coming.
Well, make sure you take a moment to celebrate that big win. And thank you so much for sharing it with us today. And I know when you're working on one of these big projects, like you said, it's always on to the next thing, on to the next thing. But we all like to encourage our listeners to take a break. And often a great way to expand your mind and give your mind a break from your own science is to read. So we love getting book recommendations from everybody we have on the show. Trudy, do you have a favorite book or one you've enjoyed recently that you would like us to add to our own reading lists?
I probably have a few. I'm not sure how to narrow it down to one. I'm kind of into these self-help and how to make yourself a better person type of books. My favorite little collection recently, I would say White Fragility, How to Be an Anti-Racist, and The New Jim Crow. I'm partially through The New Jim Crow right now. And then a couple others. One, trying to help sort of be a better boss or manager, called Radical Candor, and another called The Obstacle is the Way.
Excellent recommendations. Well, we will add these to our reading lists for our listeners out there who might be looking to improve themselves along their own journeys. And we've talked about some different opportunities you've had as a scientist and mentioned some of the different places that you've gone for your academic training. But I think as a scientist, you get so many opportunities to travel, whether it's for conferences, to work with colleagues, to present at different institutions. So do you have a favorite place, Trudy, that your science has taken you?
I have to agree. I think one of the greatest things about being a scientist is the ability to travel and see amazing new places. So I'd say the most recent exciting trip was in November. I got an opportunity to go to Barcelona in Spain. The people were amazing. The food was awesome. I got to go to some of the top world's cocktail bars and had some of the best cocktails my entire life. So that was definitely a highlight. I also really liked my trip to Germany. The bread is amazing. And also Banff, Canada, which I thought was just unbelievably gorgeous.
Awesome. So what were some of your favorite sites in Barcelona?
Park Güell, the sort of place where you get a view of the entire city in the water. My other favorite part of Barcelona was seeing the Pala Güell museum or mansion that Gaudí had constructed in the late 1800s.
Oh, that's so cool. And you mentioned Germany as well. I had an opportunity to go there several years ago. Where in Germany were you?
I kind of went to multiple places, but I was in Essen and Duisburg, and then we did a little bit of traveling outside of there. But I was visiting a collaborator at the University of Essen, and I'll just never forget the little bed and breakfast they had me stay at. And you come out in the morning and they have this breakfast buffet with all of these different unique breads that, coming from America, when I saw them, I thought they looked almost plastic. And I thought, oh, there's no way that bread can be any good. But all the other people staying there were scooping up this bread. And so I thought, well, I need to go try that. And then I tried it and I was just completely blown out of the water. Like, oh my goodness, this is what bread is supposed to taste like.
That's awesome.
I was trying to stuff it in my pockets. How many of these little buns can I take home?
Your snacks, yeah?
Yeah.
And then third, you mentioned Banff in Canada. What was the occasion for traveling there?
We had a conference there as well, and we were staying in some kind of castle. And it was kind of amazing. The weather shifted really dramatically while I was there. I got to see this one hike where there was no snow, and then I got to see the same hike covered in snow and icicles and everything. There were moose, and it was just a stunningly gorgeous area.
Well, that sounds absolutely amazing. Thank you for bringing us on these trips with you through your memories. Well, Trudy, it sounds like you've had some fantastic travel opportunities, and over the course of our conversation, you've mentioned lab members and colleagues and collaborators who you've been able to work with over the years. And I think oftentimes there's these certain reputations or certain ways that scientists are portrayed in the media. And I think the people that you meet in science often don't fit the stereotype. So we try to break some of those on our show. So do you have an example of a fun lab tradition or just like a quirky or funny memory that you shared with colleagues that shows this human side of science that may go contrary to some of the stereotypes that people may still have?
Oh, yeah. I think the JAX lab experience definitely takes the cake. We used to take a day per year where we would get into teams. We had so many people in the lab. We would form these little mini teams and we would dress up in some kind of costume consistent with our theme. And we would travel around all of Boston, usually doing some kind of scavenger hunt, incorporated with a pub crawl, incorporated with some kind of Olympic games that we had engineered. And I remember my team was the Brady Bunch, and we were doing sack races. And some of my colleagues, like David Felter, were pelting me with water balloons while I was trying to do a sack race. It was crazy. And we would do that all day and all night till we were completely exhausted. Those are some good memories.
Oh, this sounds fantastic and quite elaborate, actually. So have you adopted any sort of maybe slimmed down version of this tradition for your lab?
Yeah, I can't say that we've done anything nearly as cool as that. But one of the things we do for everybody's birthday, we'll have a little party and we have this gratitude circle where we'll go around the room and everyone will say something about the birthday person. Usually something that they like or appreciate about that person. And it's always very entertaining, very positive and uplifting. And I think it helps everyone in the lab sort of feel included and appreciated in some way.
Well, Trudy, I love this tradition. I think you're the first one to mention it. How did you come up with the idea?
Someone in the lab actually had the idea. We were always having these little birthday parties and putting the birthday person on the spot. But then someone in the lab one day just thought, we should do this. And then like, yeah, that's a great idea. And so we adopted it and been doing that consistently ever since.
Well, I think that's wonderful. And like you said, helps to foster a great lab environment where people get to know each other and get to appreciate everyone's different personalities and what they bring to the group, which I think is so important. Because teamwork is essential when you are solving big problems like working on cancer. So oftentimes there are major barriers that stand in your way. Things like funding, as we mentioned, can be a challenge, as well as just putting together the staff, the technology, and everything that you need to get these projects done. So if we took away the barriers, Trudy, that normally hold you back and you could do anything, what is the one question that you would want to answer most?
I think the question that's burning in my mind right now comes out of this recent project that we just published, where we came to believe that not only small cell, but many other cancers throughout the body are probably arising in these basal cells. And some of our unpublished work suggests that the basal cells have a unique relationship with the immune system. cancer tends to be a disease of older people, and that it takes a really long time for the genetic alterations to accumulate in people so that cancer finally arises late in life. But we also suspect that the immune system is really keeping early stage cancer cells in check for a really long time. And then suddenly there's some kind of breakdown with the immune system such that cancer can sort of emerge and take off. And our data suggests that the basal cell has this interesting relationship with the immune system. And so I think if we could do anything right now, I would really throw a ton of people and resources at understanding how and why the basal cell talks to the immune system in a different way, and trying to learn the rules of why it is that sometimes the immune system will eliminate these basal cells, and then in other cases, they seem to escape the immune system. Because I think this could have important implications for preventing cancer at its earliest stages.
Oh, definitely. So, Trudy, what do you see as the biggest barriers to, I guess, you starting this project today?
Funding. It's always funding and people. But yeah, funding, and funding.
That's right. A, B, C, and D, all funding. Yeah. But that's actually exciting and maybe heartening to hear that it's not a technological limitation, that it's not just this totally infeasible project, but it's something that you could potentially solve given extra time and funding and staff.
Oh, for sure. Where we are right now is a very exciting time because I think so much could be done. We have the tools and reagents and resources and ideas. It's just a matter of time and money.
Well, Trudy, you're giving us something to look forward to in the future. And you've shared a lot of insights from your career with me and our listeners today. I would love to end with some advice. So do you have a piece of advice that somebody gave you at some point that really helped you that you can pass on to our listeners today?
This was actually from my therapist. I saw this therapist in Utah for like 10 years, an amazing guy named Chris Robertson. And he would always say that if something comes out of someone else's mouth, it's about them. And the point was really, don't take things too personally. A lot of times we really care what other people think, and we want people to think positive things and good things. And I know, like, as a sensitive person, I can be deeply sensitive to what someone else says or thinks. And this was kind of like a reminder that what people say is really a reflection of what's going on in their mind and in their world. And it's okay to just let other people have their thoughts and be confident in your own self.
Well, I love that advice, Trudy. Is there any other last piece of advice that you would like to share personally with our listeners, or perhaps a last note of inspiration that you would like to end our conversation on today?
Not really, but I do appreciate you reminding me we need to go celebrate this paper. So I think I'm just going to use the time to go plan a party.
I like it. Listeners, the advice is celebrate the wins.
Right.
Well, Trudy, thank you so much for your time today. If our listeners want to learn more about you and the work that you and your team are doing, what is the best way for them to do so?
I would say follow me on Blue Sky at TG Oliver. And you can also visit our website. We have a lab website called www.theoliverlab.com, where you can learn more about our work and contact us that way too.
Fantastic. Well, listeners, definitely take some time to check out Trudy's lab website. Definitely get connected with her on Blue Sky, TG Oliver at blueskysocial.com. And Trudy, thank you again so much for joining us on the show. It's been such a pleasure to learn more about you and your work.
It was really my joy. Thank you so much.
Well, it was wonderful to have you with us, and listeners, always great to have you here as well. We hope you join us for our next episode of People Behind the Science.